All right, welcome to lecture 5. We are meeting here through a video. For obvious reasons, we are not in contact. And soon after this lecture, you will be able to have a conversation with me on social media. We can meet on Facebook. I'll throw this video on YouTube. And obviously, it will also be in Blackboard, as it's more convenience for everyone else who's on Blackboard, because everyone is supposed to be on Blackboard anyway. So let's deal with some logistical issues. On the 5th of September, I'm expecting assignment number 2, which I will give you in somewhere in this lecture. And remember, on the 29th of August, our semester test 2 still stand. Although in my absence, you still write this test, and you cannot find me in until in September, so I'm not available until we meet at lecture 6. All right, so let's deal with some recap. Previously, we studied allosteric inhibitors and activators, modulators, feedback inhibition. This is very, very important. So I would like you guys to take into consideration that you need to know feedback inhibition, in-depth of feedback inhibition, in-depth of allosteric modulation. Because as you know that activators and inhibitors are compounds that alter the activity of multimeric allosteric enzymes. You know how they do this. You should know how they do this, because they then bind to their own binding site on the enzyme, which is not the active site of the substrate. So this information you need to know. So if you look at this image here, you would see that there is a different site for inhibitor or activator, and there's a different site for substrate. And those are kind of things that I would like you guys to go and learn. Actually, let's make this an assignment. You guys go and differentiate between the two allosteric modulators with the aid of diagram or diagrams, whichever way you come up with. And then we'll just look at the assignment that way. So go on Blackboard if you need a proper explanation of how to write this assignment, you will find it. All right, so today we are here because we need to deal with enzyme biotechnology. Moving on. Okay, so what is biotechnology? What is biotechnology? So what do you understand biotechnology to be? So go to dictionary, you will find the term biotechnology explained. Since its inception, it was also explained. So you'll find it. So there is this exploitation of biological processes for many purposes, especially the genetic manipulation of microorganisms or plants or animals or humans for the production of whatever we need, antibiotics, hormones, enzymes, many other processes. So biotechnology involves a whole lot of processes and engineering and things like that. So they are differently in medicine, drug discovery, that is in pharmaceuticals. In agriculture, you would find the modification of plants for heat resistance, production of heat resistant crops, pest resistant crops. You would also find in industry what we will also deal with in this lecture or next lecture, the production of beverages, alcohol, food, feed, pulp and paper in industry, and you'll find biotechnology involved. And in this case, specifically, enzyme biotechnology would be involved in this industry. Also, environmentally, if you guys would go and look at how bioremediation is, and you would see the involvement of enzymes in bioremediation, although there might be some negative effect to bioremediation. Some guys have been studying how microorganisms that are used in bioremediation could negatively affect the environment, although it's positive to use them in cleaning up possibly oil spills and other waste material that might need bulk microorganisms in bioremediation. All right, so the use of microorganisms as opposed to the use of plants and animals does have economic advantages. And that's what I would like us to look at now. So obviously, economically, it's economical to produce more at a short space of time, and of course in a small production facility. And this is what you get when you use microorganisms as opposed to using plant and animals. All right. For example, during the production of a milk coagulating enzyme used in cheese manufacture, the traditional approach will be to use the enzyme extracted from the stomach of a calf. This is a young cow, whichever, still feeding on its mother's milk, right? So you would find the enzyme in its stomach. But the average quantity of this enzyme extracted from a cow's stomach will be about 10 kg, right? And it takes several months, of course, intensive farming, feeding of the calf itself in order for you to get the 10 kg. Imagine, let's say it takes about 3 months. I'm not sure how the calf is raised. 10 kg, you wait for 3 months to get to 10 kg of this enzyme that produces cheese. So everyone else is waiting for cheese for 3 months, right? And in contrast, if you were to use microorganisms, you will just take a gallon, 1,000-liter fermenter, and put your recombinant Bacillus subtilis, which is a bacteria, and within 12 hours, you are producing 20 kilograms of the same enzyme. Okay, so it's economical to use microorganisms as opposed to plant and animals. And remember, in research and in all these production facilities, ethical issues are a concern when we talk about regulations. So to use animals or plants, you need to apply for ethical clearance and all these regulations. You need to pass through all that. But with microorganisms, you do not need to do or any of that. So it's economical to use microorganisms. So a further advantage will be the ease of extraction. It's easy to extract enzymes out of microorganisms as opposed to plants and animals. And obviously, some of these microorganisms will release the enzymes into the broth or the media that you are growing the bacteria in or the yeast in, which is very easy for you to then get. You just get the broth and throw away the cells, and you have your enzyme. Just clean it up. They will produce, they will secrete the enzyme extracellularly. But with plant and animals, there's no such. Okay, there's no such. There's no extracellular production of enzymes. Okay, another economic advantage would be fewer extraction and purification steps. Okay, like I've already mentioned that the ease of extraction, which also, of course, when you're trying to extract it, you're trying to extract enzymes, you don't want to spend the whole day trying to extract enzymes. But with microorganisms, you do this and you purify, break the cell, and very simple techniques that are optimized for it. And one other thing is that you can do this in the same facility. You can grow a microorganism in the same facility. You can just move next door in order to move to a fermenter and grow it larger in large numbers, and then you can move to another room and you do your purifications. Whereas when you do it with animals, you need a farm to grow the animals, and you need to move the animals to another factory in order to do that. And plants need to be grown in the forestry, and you see the hullabaloo that comes with such. Okay, so and another thing is that when you use plants and animals, you need to wait longer times, like I've already mentioned. When it comes to plants, it might be seasonal, although there is modification now these days, but it might be seasonal. You might need to wait for summer in order to produce enzyme, or you need to wait for winter because the plant you're using would conditionally do better in winter. Okay, and there are also technical advantages to using microorganisms as opposed to using plant and animals. Okay, one of those is that the enzymes will be more stable as some bacteria, most bacteria will be stable in high temperatures, will be stable in sort of a range of pHs, you know. So the enzyme that you produce might also be stable, because you would understand that for the cells to be stable in those higher temperatures, it might be the influence of the enzymes that are stable in higher temperatures. Okay. And then it's also very easy to modify bacteria as opposed to modifying plant in animals. Okay, so you modify the bacteria that you are looking into expressing your enzyme in, producing enzyme, purifying enzyme from that bacteria. You might need to genetically modify it, and it's easier. That although some people have expressed proteins and enzymes in tobacco, for instance, which they genetically modified, but we remember, for you to do that, as I've already mentioned, you need a very big facility to grow a whole lot of tobacco. You need a farm, you know, to produce it in bulk. Whereas you need a drum or just a bigger container, you know, to grow your bacteria with. You just stirring, like the way they produce traditional beer. You understand what I'm trying to get to. Okay, so let's look at a biotechnological process for enzyme production. This could also apply to production of other molecules such as proteins are enzymes in any case. Okay, so you would need to do some sort of screening for you to look for that bacterial species that will produce that enzyme that you're looking for. Okay, so you cannot just say, for instance, you need to produce an enzyme that will help in the manufacturing of cheeses we've already mentioned. You cannot just go and isolate by any other bacteria. Say, for instance, you just go to a cow and you swab its mouth, and whatever bacteria that comes from there you grow. No, you do the screening. If you were to start this process, you might have to do screening of a whole lot of bacteria to see from the stomach of the cow to see which one does that. But a good thing is science has been here for a long time, and most of these have been studied, so you know which microorganisms to go for. But in that species that you already know, you also need to screen within that, because if you grow it in a petri dish, if you grow the microorganisms in a petri dish, you will see single colonies, and then microbiologists will tell you that this colony will be able to produce the enzyme better than the other colony. All right. But then after screening, of course, if there's need for modification and then you do the modification, molecular biology will tell you that the possibility of genetic engineering to just produce the microbial strain in order to then get your optimum enzyme production. Okay, so still in optimization in the lab, you will produce it, you will grow and express this protein or this enzyme at a small scale, which will be a laboratory scale pilot. And you produce and then you purify, you see the enzyme is there and all that. Then you move into a pilot plant, which will be a little bigger fermentation, in order for you to see that you can, is it possible to produce it in bulk? If that is then possible, an industrial scale fermentation can then be used, and you go commercial on that. And economically, this is when there would be sale of enzymes that goes into other industry, it goes into your research, and there'll be job creation in this process. Okay, we've already mentioned the petri dish, the flask, the large fermenters, and the removal of cells. Of course, if you're producing enzymes extracellularly, you need to remove you need to separate your cells, and you can do this by different processes that we are going to look at when we look into purification. And of course, there's also refining step where you concentrate your protein, because you don't want to carry a fermenter to go and sell the enzyme. You need to concentrate it, purify it, and modify if there's need before it goes into selling. So you would see as we go along that you might need to sell this enzyme in liquid form, it's insoluble form, I mean to say, or you might need to sell this enzyme in dry form, in powder form, I mean to say. All right. So although there's been problems with selling enzymes in powder form, some enzymes you are just forced to sell it in powder form, although it's a bit dangerous to do so. But then when you sell it in powder form or liquid form, there are processes that you need to go through in order to change from liquid to powder or powder to liquid by reconstitution or by waxing and things like that. Okay, so protein engineering before production. What we're trying to get to is to show you that these enzymes, whenever the microorganisms are being engineered or have been engineered, they are used in several industrial processes. Maybe this slide might not be that, but you will see alpha amylase, the very first one, is used in beer making, in alcohol production, in starch hydrolysis. And if you look at the traditional beer making, you go and ask at home how they do it. You will see that some of these people who are doing it in the villages don't even know that this is starch and this is amylase, that is, this is yeast that produces amylase. They don't even know these things, but they have the science. Okay, so let's look at where most of these enzymes come from. The source, the bacterial form, or the yeast form, human form, animal form. Okay, let's just look at some of them randomly because it's a whole list. We can't go through everything. So look at the acid proteases that comes from Aspergillus niger, and that's where you see your alpha amylase in Bacillus, the Bacillus subtilis. If you go down, you will see penicillin acylase from E. coli, and you would remember penicillin as an antibiotic. It's now becoming, most bacterial species are now resistant to penicillin. Maybe the engineering of such enzyme could help in that. So in some of the analytic reagents, they used glucose oxidase. It's found in Aspergillus also, luciferase, which is also found in marine bacteria or firefly. The firefly, you remember the firefly that glows in the dark. So you find, if you want luciferase as an enzyme, you can find it from the firefly. Peroxidase, horseradish peroxidase, that is used in Western blot. You see lysozyme found in hen egg, chicken egg, and your DNA polymerase, which is found in bacteria, Thermus aquaticus. That was first isolated in geysers, like high temperature water sources. Okay, so that's the reaction. Might just pass that because we can talk about that later. Okay, so the application, you would see your as we mentioned, acid proteases in milk coagulation, in cheese manufacturing, like your renin enzyme will be involved there. Alpha amylase, that is found in Bacillus. Conversion of starch to glucose or dextrans in the food industry, like in beverage manufacturing. Penicillin acylase in E. coli is used in the 6-APA formation for production of semi-synthetic penicillins. You see, as I was talking about the production of penicillin using penicillin acylase. So, for instance, if you do modification or engineering of penicillin acylase, there's a possibility that we might come up with penicillin 2 or penicillin 3. That would be more effective than the normal penicillin that we know. So in analytical reagents part, you would see, I've mentioned luciferase, that's found in firefly. Bioluminescent assays involving ATP. So even in the determination of glucose, luciferase can be used, and this is in diagnosis of what? Diabetes, for instance. Okay, so your lysozyme will be used in disruption of mucopeptide in bacterial cell walls. So if you need to open up a bacteria If you need to open up bacteria, for instance, bacteria in bacterial culture, you need to open up bacteria in order to release the enzyme of interest. You would use another enzyme, lysozyme. You see, so this is where it's important. And DNA polymerase in PCR, the DNA amplification, you find your tech polymerase, which is very, very popular in PCR. It comes from Thermus aquaticus. And the application right there is PCR in molecular biology. So you cannot do PCR without an enzyme that helps you build double-stranded DNA. Okay, but you also need to make sure you improve certain things when you doing your enzyme production or the engineering. So factors such as kinetic properties of enzyme turnover, like your KCAT and your KM, which determines catalytic efficiency of the enzyme itself. That needs to be improved. So after modifying and improving your enzyme, you measure that and you see that you're not up to where you are going to. So you need to improve your enzyme, your organism, in order to improve your KCAT and your KM. All right. So the other thing that you need to look at is the thermostability. If your enzyme and enzymes you're producing can't even survive at 37 degrees Celsius, for instance, or 42 degrees Celsius, so there's need for you to continue improving. Okay, the stability and activity of enzyme in non-aqueous solutions or solvents. In case you want to, for instance, wax your enzyme in powder, because you don't want to transport it across length and breadth of continents in solution or in liquid. You want to make it powder. You need to make sure that it can be stable in powder form and it will still be active when it's reconstituted. If that is not the case, then there's need to continue improving. Specificity. If your enzyme it cannot be specific to the substrate that it's meant for, it needs to be improved. Cofactor requirements. You need as little as possible cofactors. So we improve our enzymes to be able to function, possibly without cofactors. So we don't need a whole lot of things in order to get our enzymes to be active. But if they can use cofactors that are readily available, then we improve our enzymes towards using readily available cofactors. And the pH. Usually, most of these enzymes will survive very well between neutral and alkaline rather than acidic. So you need that optimum pH to be around normal, like you get what I'm trying to say. And you also need to make sure that the molecular weight and subunit structures are intact and they don't just break, affecting the functionality and the activity of your product. Okay, so there are several steps that are needed in enzyme engineering or enzyme biotechnology, for instance. We've already mentioned isolation, but then when you isolate, you need to then remove that DNA from what you think or what you have confirmed to be the best production strain of the product that you are looking for. Isolate it, screen and isolate, put it aside, remove the DNA from there because that is your fingerprint. Remove the DNA from there. Keep it for future use. So in case when you need that product again, you retrieve your DNA, put it back into another bacteria or the same bacteria that does a better job, you continue producing in the future. Okay, but that DNA, the cDNA that you get from your strain that you've isolated, it needs to be sequenced. In a way, we are trying to say you need to keep the information of that DNA in case, okay, say for instance, you keep your DNA and then you lose it. But if you have a full sequence of that DNA, you can synthesize it, or someone can synthesize it for you. There are experts out there, and then you can get that DNA back, all right? I'm sure you guys, you are learning how to use the snap gene in the other module. You're learning a whole lot of things, which is very, very good. So most of the things that we are talking about here are not completely new to you. Okay, so after producing the protein or the enzyme, it needs to be crystallized. You need to learn the structure. Secondary structure is tertiary structure. And then you need to be able to say, this is how it looks. This is the structure of the protein. These are the components. These are your alpha helices, your beta sheets, and this is how it's structured. Because then when you take your sequence, you need to retrieve your sequence, and you then continue producing at a later stage. You do the same, you do crystallography, and you see the structure is still the same. Then you know that we can continue using the protein for the same job. Okay, so sorry guys, you will not be able to stop me if I'm too fast, but then make notes, then you can ask me questions when we're done. Okay, of course, if there's need for improvement, that's where you introduce your mutagenesis. Like if you want to produce a mutant out of the sequence, you can then introduce mutagenesis. That can also be done when you want to then change things around in the sequence and to produce a desired target product or your enzyme, which you can then test for that improvement that you're looking for. And the testing will involve techniques such as an assay, an activity assay, for instance, which is quite a standard procedure to do to test whether your enzyme it's good enough, because then the functionality will be seen in activity. Of course, we spoke about crystallography in the NMR. You can do your CD spectrophotometer spectroscopy. You can do your fluorescence spectroscopy. There's just a whole lot of techniques that you can look at your protein to see if it's still the same. Okay, so still on the same issue, we need to look at molecular evolution of this protein, which we can manage as scientists. Okay, but then what is molecular evolution? This is a process of change in the sequence composition, the sequence that we just spoke about, that you have extracted the cDNA, sequence composition of a cellular molecule. This is in reference to DNA, RNA, it could be a protein. You can get a sequence of a protein, or RNA or DNA. Okay, so there's a process of change in the sequence of such molecules from one generation to the other, and that needs to be monitored. That molecular evolution needs to be monitored. That's why we do our screening and isolation, because along the way there might be changes in molecular evolution, and you find that the bacteria that you know used to produce certain protein or enzyme no longer does that. So if you look at the scheme that's on the screen, you would see where you start in clones in selective plates, where of course you need a selective agar that will grow that bacteria, that organism that produces the enzyme you're looking at. Your enzyme expression, you look at whether the enzyme can still be expressed from that microorganism, and you do screening assays and you pick that one that is best, and then you move it into the next generation. It now becomes the parent, and when the cycle continues, you will find a different parent. Some of this will be losing viability in the production of that enzyme, and the cycle will continue like that. Okay, let's look at some technicalities in enzyme purification. Okay, so within the cell, enzymes are generally found along with other proteins, nucleic acids, polysaccharides, lipids, and the activity of the enzyme in relation to the total protein present, that is your specificity activity, that can be determined and used as a measure of enzyme purity. All right, so a variety of methods can be used to remove contaminating material, and that is in order to purify the enzyme and increase its specificity activity. So hence we are talking about enzyme purification. So we need to purify this and that enzyme. So the enzymes that are used as diagnostic reagents in clinical therapeutics that you see, you know, are normally prepared to a very high degree of purity. And because great emphasis is placed on the specificity of the reaction that is being catalyzed. Clearly, the high level of purification, purified enzymes are needed due to that. Okay. All right. So we'll probably skip this screen because this is this slide, because we've been talking about that. So this is just a repetition. Because it's the same screening, isolation, modification to enhance enzyme activity, and the protein structure and function depends on folding. That's or misfolding or dysfunctional protein. When protein misfolds, then it becomes dysfunctional, and its activity is then hindered. So let's continue on purification. So right after you have grown your bacteria in fermentation, microorganisms rich in the required enzyme you need to then cool that culture rapidly down in ice to, well, not freeze it, but like 4 or 5 degrees Celsius. This is to prevent any further microbial growth and also to stabilize the enzyme product. So that's hence you see most of the molecular biologists and biochemists, and they'll be working on ice in most cases just to make sure that everything is cooled down and the temperature is kept down. You can also monitor and change the pH right after the growth of that, the microorganism from fermenters or from your flask, whichever part you are dealing with. So that's part of pretreatment. And of course, if you are growing or you are looking to produce enzymes in yeast, you might need to remove it by centrifugation. If you're growing your enzymes from bacteria, you might need to use aluminum sulfate. You just flocculate, or calcium chloride to flocculate the bacteria, which then cause clumps, and then the bacteria will come out of suspension. This could also break the cells. So moving into treatment, extracellular enzymes are found in the liquid component of the pretreatment process. Like we have mentioned, that it's the ease of extraction with using microorganisms makes it an advantage, because you're not going to go too far. So in pretreatment, you might be able to collect your enzymes right there before you. But intracellularly, enzymes that are produced intracellular will require extensive treatment, like your breaking of your cell wall and then continuous and separation of those enzymes. Okay. So let's move into looking at the process of rupturing cells, still in treatment. So you might need to use ball milling, and glass beads are involved here. This is, you mix your glass beads. It's like crushed glass. So you mix your glass beads with the microorganisms. Of course, crushed glass will then physically cut the cell wall of your bacteria or your yeast, to then remove the components within the cell. That will include your intracellular enzymes. Or you might use lysozyme, as we've already mentioned, that lysozyme that is produced in hen egg white can also break your cell wall, releasing the components of the cell, including your enzyme. Free surfactals, using liquid nitrogen or minus 80 degrees Celsius freezer. Of course, what you need to do, then you immediately freeze, immediately thaw. That will also influence the breakage of your bacterial cell wall. And in French press, where high pressure is used, in osmotic shock, and also in sonication. So if you look at number 2, number 3, and number 6, this is what we can be able to readily show you in our laboratory. That you can use lysozyme. We have done that a lot, and we continue to do that. Freeze thaw cycles, we have used liquid nitrogen, we've used minus 80 degrees Celsius, and your sonication, that is being done in the lab currently. That just through that vibration, you break the cell. Okay, so still in treatment, after breaking the cell, obviously your intracellular enzymes or membrane enzymes, proteins, they are in solution. But then they are not in solution alone. You need to then separate non-enzymatic molecules from your target enzyme. And you need also to separate your target enzyme from other enzymes that you are not looking for. And there's a sequential fashion that you can follow to do that. There's size exclusion, hydrophobic interaction, ion exchange, affinity, reversed phase. But we'll just look at size exclusion, ion exchange, and affinity. That's enough for you to learn how you can separate enzymes from non-enzymatic molecules or from other enzymes that might interact and mask the production of your enzyme. Okay, so let's look at separation of these enzymes by salting out. For instance, before you go into processes such as gel filtration and all, salting out would, okay, let me read what I have here for you. Proteins remain in aqueous solution simply because interactions between the hydrophilic amino acids, these are water-loving amino acids, and the surrounding water molecules. So there is interaction between your water-loving amino acids of the enzyme. And then that is why your enzyme now is in water and is happy. But if you put in a salt, you then reduce the water molecules because the salt will then react to your water molecules. And that is simply by increasing the ionic strength, of course, of the solvent, right? By adding ammonium sulfate, which is a salt that we are talking about. But when you remove water molecules that will then interact with ammonium sulfate, you leave proteins to interact with each other now. And then when they interact each other, they form clumps, which are called precipitates, and then it comes out of solution with that. So it's easy. I've been telling other students in the lab that you can easily see precipitates. Look in the tube, you will see precipitates, and you know that your protein is no longer in soluble form. When you see precipitates, you see clouds inside the tube. You know that your protein is then clumping with each other. Some proteins will simply clump with each other because you have frozen them, for instance. Sorry. So your protein, your precipitate, which contains your enzyme of interest and other proteins, can then be filtered or centrifuged or separated in order to separate them from the supernatant. Okay, so the other way to do this is fractional precipitation. Okay, since different proteins vary in extent to which they interact with the water, this is before you add an agent such as ammonium sulfate. It is possible to perform fractional precipitation using a series of additions of ammonium sulfate, which then increases the ionic strength in a stepwise fashion. That by doing that, you are removing the precipitate at each step. You remove precipitates. Okay, and that simply means that if you're doing it by increasing the concentration of ammonium sulfate, a certain concentration will remove certain precipitates. Not every other protein can be removed at any concentration. So you do it stepwise. You are removing certain enzymes or proteins at each step. All right, and subsequently, this is when we get to techniques that needs to separate your enzyme of interest differently. It could be your ion exchange chromatography, which is effective during the early stages of purification, right from fractional precipitation. You need to get to ion exchange because then you still have a mixture, although you have sort of removed some of these things along the way. But if you get to ion exchange, which is often effective during the early stages of as we have already mentioned, the protein solution, this is how you do it. You add your protein solution to column containing insoluble polymer. And in this case, let's say the polymer you have, it's cellulose. The stationary material that you pack your column in. It's cellulose, right? And this has been modified so that its ionic characteristics determines the type of mobile ion that binds to it. In which case we're talking about the charge that binds to it. So protein whose net charge is opposite to that of the ion exchange material will then bind. So if your protein is positively charged, the material is negatively charged, your protein will bind to it. And then all the rest of the proteins that are of the same charge would then pass through, right? So if you increase the pH of the solution, you alter the electrostatic forces that it's allowing your protein to bind to those polymer or the cellulose material in the column. And when you alter those forces, your protein then is released and you are able to collect that enzyme or that protein. And this is how ion exchange works, right? So if you look at the image that I'm showing now, you would see that the resin, the bigger gray particles, are your negatively charged resin, and the blue are your positively charged protein. And the negatively charged protein that are shown in I don't know what color is that, those are flowing past, but your positively charged enzymes or proteins are then binding to the beads. Okay, so you would see. So the gel filtration, which is also called size exclusion, because then your molecules are excluded according to size in this case. So gel filtration is also called size exclusion. So gel filtration can be utilized in the later stages of purification protocol to separate molecules on the basis of their molecular size. So columns that contain a bed of cross-linked gel particles. So the particles are then cross-linked. Okay, the particles that are stationary packed in a column, they're cross-linked. All right, so there are types of that, many. You can Google them up, you'll find your Cephadex, your Cepharos, there are many. But these gel particles, they exclude large protein molecules while allowing the entry of smaller molecules. So this is what happens. So if you put in a mixture of crude enzyme through the gel, the smaller particles of your enzymes will get into the gel, because then the gel forms matrix, but those smaller ones will get trapped into the gel, and the bigger particles will not find anywhere to get trapped, and they will find their way past the gel, and they'll be excluded. So your large particles will come out first, but your small particles will be trapped within the gel. But with time, they get trapped and they get released. By the time they get trapped and they get released to get out, your larger particles are already out of that column. So this is the principle of gel filtration. If you look at it, you would see. So look at this image here, you would see. You'll see that in a mixture of 3 proteins, the bigger ones are then released out. So if you look at, let's get a pointer. So if you look at the last column here, you will see there are sort of blue and red dots that are circled in. So these are your enzymes that are trapped. So these are smaller molecules that are trapped within the gel, while bigger molecules, such as the ones that are shown in yellow, are passing through because they don't have a space to get trapped. So they are happy, they are passing through. So when they pass through, the second part of the mixture of 3 enzymes, the second size, is from bigger to medium to small. So the larger ones will come out, medium will follow, and the smaller will follow, just like that. But there's also another technique called affinity chromatography. I believe that what I'm teaching you today, it's something that you have already learned in other techniques. So those of you who have done techniques 2 will remember this kind of thing. So we are repeating this, specifically looking at the purification of enzymes in enzyme biotechnology. Okay, so it's known that affinity chromatography, it's also a one step. So you can get from crude, you can bring your cell, get from crude, take out the components and throw them in affinity resin. You can still get 95% pure protein, but it's always good to go through a sequential fashion of purification from your ion exchange, your gel filtration size exclusion, and getting into affinity chromatography as the third step, because then you have excluded a whole lot of crude stuff, and then you do not really burden your affinity chromatography or your affinity resin at this stage. Okay, so how does this one work? The column is packed with a stationary phase in which a ligand ends a molecule, such as your substrate, for instance. So let's talk about a substrate, and let's not talk about inhibitor and cofactor, but those can also be used as ligand. So you pack your column, you put your ligand, you know that the ligand will bind to your enzyme. So once you add your enzyme mixture or you pass it through the column, your enzyme will bind to the ligand. In this case, we talk about substrate, inhibitor, or cofactor, any of those. As long as you know that the ligand will bind to the resin in the column and will also bind to your enzyme. So you pass your enzyme bind. Now you have a combination of three: your resin, your ligand, and your enzyme. Right. So if you increase the concentration of your ligand, you will then release the ligand that is bound to the enzyme from the resin, because then more ligand is coming through. They also need to bind competitively, pushing out the other ligands that were bound, and you can then collect your enzyme. Or in another case, you can increase you can also increase the ionic strength like you would do with ion exchange. But then you should use a salt or a compound that competitively binds to the ligand as your protein would or your enzyme would. So when that comes in, it pushes out your enzyme so that it can bind the ligand competitively. By then doing blessing in disguise, you then collect your enzyme. Okay, so it's okay, let's get the cursor out. All right, so there are many alternative affinity chromatography procedures that exist. One of that is that you can do your tagging when you know, for instance, let's talk about GST or HISTEC. But okay, let's talk about HISTEC, because then I've used HISTEC before, so I'll be able to explain to you properly. Okay, so the molecule that you have, through engineering, you can be able to add amino acids. If you add 6 or more amino acids to your histidine, polyhistidine amino acids to your molecule, those histidine tagged that histidine tagged molecule of yours will be able to bind to nickel, for instance, as a ligand. So the histidine part of your sequence will be able to bind to the nickel. So you need to use nickel in the resin so that your protein or your enzyme will bind to the nickel. But nickel also binds to what? To imidazole. Okay? So for you to get your protein out that is bound to nickel in the resin, you just put imidazole. Of course, high concentration of imidazole, which will then displace your enzyme or your protein out of that binding relationship. Then you can collect it. All right? Okay? That's it. So we use nickel NTA agarose resin. So read around it, you would see what we are talking about. So look at this scheme here. We can talk about just GST, we can talk about polyhistidine protein. Okay, with GST, what then happens is your glutathione is attached to the beads. Your protein of interest attaches to glutathione. You add more glutathione, displaces your protein. That's how simple this is. But with polyhistidine tagged protein, what do? It's exactly what I've explained to you, that your resin comes with nickel, or you can also bind nickel yourself to the resin. Your protein, which has histidine amino acid molecules that you have modified or engineered attaches to the nickel. You increase imidazole concentration, you get your protein out. All right? But then you don't want to keep imidazole in the column for long. So you then push imidazole out. You can use a whole lot of compounds. You can use sodium hydroxide, for instance, to remove imidazole from binding to your nickel. If you use sodium EDTA, you will remove your imidazole and you will also remove the nickel, in which case you need to put nickel back for subsequent purification. All right? But then when you're done purifying the enzyme through the steps we just talked about, like we started with salting out, your ion exchange, size exclusion, affinity chromatography, and then let's say you now have your 99% pure protein, but you then need a finished product. Okay, so what you need to look at, which is quite important, I think I've mentioned it along the line. What you need to look at, which is quite important, is that if you are getting your enzyme out in liquid form and you're supplying them in liquid form, that is okay, and it should be okay. But then look at the pH, look at the ionic strength of the solution. Usually you find that these proteins are also, for them to be, for the shelf life to be increased, you can also add glycerol, because then you mask it and you protect it from a whole lot of changes while it's being transported. But then if you need to supply this enzyme, let's say you really need to supply this enzyme in powder, like gallons of powders that that moves around from industry to industry, from production to application. What you need to do is to avoid sending it out as dust particles. So that way you can use a process called prilling, which is mixing the enzyme with polyethylene glycol, PEG, also called PEG, and you prepare small spheres. So you just increase the size from the smallest size you have to around 0.5 millimeter, for instance. Those kind of particles will not just easily fly around. Okay, so nobody will breathe them and get allergic and all sorts of things. And then you protect your enzymes within a clamp. But make sure that these have been optimized in a way that when you reconstitute them in the proper buffers, they will still be active. Okay, and their secondary structure will still be intact. There's also a process called marumarizing, which is a mixing of enzyme with a binder and water. And then you wax it out. You put a wax coat around it, and then this enzyme can be kept longer and could be transported from here to there. Okay. For interest's sake, there are a whole lot of products out there that comes with the name enzyme on them, and some of these products are don't necessarily contain enzyme within them. They just probably probiotics that don't really have that enzymes that are referred to as enzymes. No supplements that just regulate the stomach pH, your sodium hydrogen carbonate, for your enzymes to work properly. So your natural enzymes that are within you, they might not be in the products that you see, but the product would be able to regulate the stomach, your acidity in the stomach, so that the enzymes that you have can function properly. All right, I think we should end here for today. Of course, don't forget to immediately after this, watching this video now, I'll give you about 30 minutes on Facebook. If you can also interact with me on Blackboard, I will also look. But let's just focus your questioning on Facebook. You can then go back to and look in the slides and ask me questions, or also look in your notes and ask me questions while I'm still active and around this subject, because later on I might just refer you to go and look at the slide again and come back to me. So now that we're still active, let's look at it together. So the next time we meet, we'll start with single enzyme nanoparticles, one of my favorite topics, and then we can move into industrial enzymology. We should be able to complete this model in the next 3 lectures or so. All right, good luck. Let's meet in the comment section.